Let me just give you an overview about what we are doing in the field of fMRI drug-cure reactivity, which is one of the really exciting fields in the field of addiction medicine and effective neuroscience in general.
This is the pipeline of the study, and well, it's important to have a clear and explicit research protocol for an FDCR study.
This is the block diagram of our model. First, we calculate the average signal of the whole brain.
So we conducted a systematic review with this aim to gain a broad overview of the main information of the fMRI studies utilizing cure reactivity inside the scanner, or so-called FDCR studies.
This is the final checklist came out of this process of Delphi study design, which has the goal of acting, as I said, as a standalone tool for describing methodological details considered to influence results of FDCR studies.
The checklist has been developed after a global expert consensus making process, and it's available. I encourage you all to read it, whether you are planning to conduct an FDCR study or if you just want to be able to read it, an FDCR study better, and to have a sort of a relatively objective tool to assess its reporting quality.
There are potentials for developing tasks, thinking about how we can develop new measures, network level measures, different levels of measures, and having pre-registered analysis pathway, as Sarah mentioned, in terms of making sure that we are looking into areas that are gonna be more reliable and have significant activation. Seeing how we can find reliable and replicable results. And as Mehran has mentioned, thinking about validity, whether it's diagnostic, predictive, or treatment monitoring validity, is another really important area in this field, and then developing new interventions.
Thank you everyone for joining us for the fMRI drug-cure reactivity in addiction medicine panel. I'm Arshia Sangchuli, medical intern at the Tehran University of Medical Sciences and researcher at the Iranian National Center for Addiction Studies. I'll be co-chairing this panel with my dear colleague, Dr. Hossein Mohaddis Ardebili. Hossein is a psychiatry resident at the Mashhad University of Medical Sciences and has been involved with a variety of projects in psychiatry and the cognitive neuroscience of addiction medicine. Dear Hossein, are you with us?
Okay, thank you very much for introducing me. I'm really glad to be here sharing this session in collaboration with you. I hope this would be a nice scientific session for all of us. I would like to greet all the audiences, and I just want to thank Dr. Ikhtiyari as my respected scholar who invited us to take part in this panel. I just wanted, before we start the panel, I think we might be in short of time in the near future, so I will make short my speaking. So I just wanted to ask the audiences, in case they had any question about any of presenters, please kindly write your questions during the session. We have something like 1 hour for this, 1 hour and 10 minutes for our presentations. We will feature 7 talks, and after that we would have likely 10 to 15 minutes talking about your questions and wrap the session up. So please write your questions, and the one who you want to answer your question in the chat space, I will wrap them up and we'll ask the presenters after the end of talks, actually about more than 1 hour from now. So the floor is yours, Arshia.
Awesome, thank you, dear Hossein. So without further ado, we can move on to the presentations. Our first speaker is Dr. Hamid Ikhtiyari, who I'm sure requires little in the way of introduction for most of you. Dr. Ikhtiyari is an associate investigator at the Laureate Institute for Brain Research and is a volunteer faculty member at the Department of Psychiatry in the University of Oklahoma. His research interests are diverse and include various aspects of the cognitive neuroscience of addiction medicine and the discovery of novel, clinically relevant biomarkers. Yeah, I'm also honored to note that he's been my own mentor for the past couple of years. He'll provide us an overview of the fMRI drug reactivity research space, its theoretical foundations, and the aspirations of the field. Thank you, Dr. Akhtiari. The floor is yours.
Hello. I'm really happy that I'm here, and thank you, Arshia and Hossein, for putting this together. I'm really excited to see young, brilliant minds are kind of moving forward in this field and putting together lots of really good activities. I'm really hopeful about this field, seeing those people who are going to be leaders in this field for the next couple of years. So for those who are newcomers to the field, let me just give you an overview about what we are doing in the field of fMRI drug reactivity, which is one of the really exciting fields in the field of addiction medicine and affective neuroscience in general. To start with, let me just show you one figure that I really like. I'm not sure if you are a great fan of chocolate cakes, but those who really like chocolate cakes, they know that when you see chocolate cakes, there are specific brain activities happening inside your brain. Any emotionally and motivationally salient cues that you see around you, like a chocolate cake, could activate some certain brain activations, some certain brain function and cognitive processes inside the brain. And the question is, okay, how we can measure these activations? How we can measure these cognitive processes involved in seeing an emotionally and motivationally salient cue, and how we can study that? To be able to do that, what we technically do, we can compare these salient images with images that are not salient and trying to find specific brain functions that are unique to emotionally salient cues. For example, when we see a stack of rocks, we still have visual activations, and we have visual activations here as well. And when we subtract them, let's say, kind of just do a subtraction between these two sides, these two sets of visual cues, then the visual activations would be gone to some extent. And then we can, let's say here, if we have a specific good memories regarding the visual cues, those activations would remain in the brain, and you can do the studies over them. So that is how we do what we call fMRI cue reactivity studies. So we try to have what we call craving related cues and neutral cues, and we try to compare them, trying to find the specific brain functions that are unique to craving related cues or salient cues that are able to activate certain functions inside the brain. As you can see here in this figure, we might have cues that are not salient for most of the people who are not exposed to drugs, but the cue that you can see on the right side of the screen could be activating certain functions among those who are heroin users. And these certain brain functions are the target for treatment, and they are target for different sorts of interventions. And thinking about how we can modulate them and how we can measure them in the process of fMRI drug reactivity is one of the most important targets in addiction neuroscience, and trying to see how we can explore these certain activations. But what are these activations when we expose people to drug-related cues? And thinking about how we can separate the activations that we have in the brain to certain brain circuits that are involved in processing cue exposure. When we are getting exposed to these salient cues, what are the activations that we are expecting to see? One of the things that we have engaged in this process would be circuits that are involved in what we call executive circuits, in executive functions, because of trying to control the level of craving that we have. So whenever we are exposed to something that is salient, there is always a level of attempt to control that. And when we try to control the desire and the motivation that we have for these salient cues, we can have activations within what we call executive circuit, areas like dorsolateral prefrontal cortex and inferior frontal gyrus. And then we can have specific circuits that are involved in what we call saliency processing. It means that how much this cue is important for us, or what are going to be the consequences of, let's say, eating a piece of chocolate cake. When we start to think about should we do that or not, what are the positive and negative consequences of taking a piece of chocolate cake, then your saliency network would get activated. And then we have certain areas in the brain that are playing a role in what we call saliency processing. Areas like inferior parietal lobe, dorsal anterior cingulate cortex, anterior insular cortex. These are the areas that are involved in saliency circuit. We also have areas that are involved in what we call self-directed circuits. The circuits that are involved in putting ourselves in the context of that specific drug-related or food-related cues and thinking about, okay, if I would be in this specific situation, how should I behave? What would be the activities I would do on that specific situation? And these are the what we call self-directed circuits, areas like dorsomedial prefrontal cortex and posterior cingulate cortex. Of course, there is significant activation within areas that are related to memories. When you have good memories with these drug-related cues or food-related cues, you have activations in memory-related circuits, hippocampus, parahippocampal areas. You can have activations within what we call habit circuits, the areas that would shape the way that you behave when you are exposed to these drug-related cues, whether it's about eating them or trying to find drug and use the drug, whatever motor functions or wired motor functions that you have inside your brain, they would get activated. Areas like dorsal caudate cortex, dorsal caudate nucleus, or putamen would be involved in that process. And then, of course, at the end, what we call gain and loss circuits, things related to reward and punishment, would get activated. Areas like nucleus accumbens, ventromedial prefrontal cortex, amygdala. So the interesting thing about drug cue reactivity is we can tap into all these networks I'm showing right now. And this is a unique opportunity for us to be able to evaluate different really important brain functions in not only drug addiction, but also anything related to how we are being as human beings. The specific characteristics that we have in terms of decision making, reward processing, executive control, self-directed processing, memories, all these things could get activated in this process. And that is the unique beauty of this field. And I'm gonna stop here. I have a couple of other slides that I would like to share and show, but if we have time at the end of the panel, I will discuss about some of the ideas that we have, how we can use this unique opportunity trying to find new treatments for drug addiction, and not only drug addiction, any other areas in kind of human behavior that are involved with self-control and what we call free will, and how we can decide between different scenarios, trying to make a better life for ourselves and others in the society. So we can discuss about those technical details. But I'm really happy that we have speakers from different aspects of drug cue reactivity, and I hope that they would give you a really good overview about different potentials within the field of fMRI drug cue reactivity. And those who are interested to join us in this collaborative partnership that we already have, trying to push forward the area of fMRI drug cue reactivity, you definitely would be welcome to be in contact with Arshia and Hossein, and we definitely would be interested to help you to get into this field. And if you like to kind of do research or follow the things that we do, we definitely would be happy to help. Arshia, the floor is yours.
Thank you, Dr. Ikhtiyari, for your fantastic introduction. So moving on, our second speaker is going to be Amir Hossein Dakhili. He's a medical neuroimaging graduate student at the medical physics faculty of the Iran University of Medical Sciences. And to give you a feel for what's possible within the diverse field of FDCR, he will be presenting a project with which he's been involved, which made use of a mixed cue reactivity and response inhibition task. It's a very interesting and increasingly recognized as important paradigm within the field. Dear Amir Hossein, the floor is yours.
Thank you, Arshia. Thank you for having me. Well, my name is Amir Hossein Dakhili, and I'm going to be speaking to you today about integration of go-no-go tasks with fMRI drug cue reactivity in people with methamphetamine use disorder. Well, to begin, it's important to know that FDCR is a very complex paradigm and can be modified to investigate a variety of cognitive functions, and also can be combined with other experimental paradigms to modulate various functions. For example, in this research, we look at combination of FDCR and response inhibition. As you know, one of the most central aspects of addictive disorders is the inability to resist urges to take drugs or other impulsive behaviors. Well, traditionally, this deficit has been investigated by simple response inhibition paradigms such as go-no-go task. This project was aimed at uncovering the brain mechanisms underlying the differences in brain processes during response inhibition in addicted individuals when they're faced with drug cues versus neutral cues. We also investigated the effect of being faced with negative emotional cues and the brain processes. Now let's take a brief look at go-no-go paradigm. Well, the way the go-no-go task helps us observe response inhibition is by showing participants a mixed go and no-go cues. And when they view cues, they have to press a button, and when they see no-go cues, they have to inhibit their impulse to press a button, and they have to inhibit their impulse responses. There is relatively scant literature available on factors that influence response inhibition. For example, we do know that when individuals with addictive disorders are viewing drug-related cues, they have more trouble inhibiting their impulsive responses. But there is little literature available on mixed go-no-go cue reactivity task using fMRI, and we tried to combine go-no-go task with fMRI drug cue reactivity. So as you can see on this slide, for this project, we used data from 62 methamphetamine users, and all the individuals were male and right-handed. We also recruited 23 healthy controls to validate the cue reactivity aspect of our task. And also you can find the inclusion and exclusion criteria on this slide as well. So this is the pipeline of the study, and well, it's important to have a clear and explicit research protocol for an FDCR study. As you can see, the first of our protocol, all invited participants were interviewed and assessed by 2 expert psychologists, and before MRI scanning, several measures were administered. And collected data included demographic information, mental status examination, clinical assessments, including drug use profile and treatment history, and also risky behavior profiles. And the participants were asked to complete PANASM VAS tests pre and post scan, and after a short break, they were asked to complete recognition task. And the scanning session also contained T1, field map, and EPI image acquisition. Well, as you can see here, the task had 4 conditions, including blank, neutral, negative, and drug-related cues, and a geometric shape on the background. And we asked participants to withhold their response to the cues with circle background as our no-go stimuli. We also know that being faced with stressful and negative cues can affect response inhibition and brain processes involved in response inhibition in addicted individuals. So we also had blocks containing negative emotional cues. So the go-no-go task used in this study consisted of 4 rounds, as you can see here, and 4 blocks in each round, and each block contained 24 trials of the same condition. The order of the blocks was pseudo-randomized in each run, and the duration of presenting each trial was about 1 second, followed by a half a second jittered interest stimulus interval, and total block time was about 36 seconds. So here you can see a schematic representation of the different conditions in our task. Participants can successfully press the button on Go Trials, successfully inhibit their motor response on No-Go Trials, or maybe they fail to press the button on Go Trials, which we call omission error, or fail to inhibit their impulse to press the button on No-Go Trials, which we call commission error. Each of these 4 conditions can happen in each of the 4 Q blocks, as shown below. So as you can see here, our results will be separated in 2 parts. Here you can see the block analysis results. We created healthy controls to make sure that the cues we're using in these tasks are valid and elicited different, differential response inhibition activations in the addicted individuals compared to healthy controls. And many of the differential activated regions, such as left superior frontal gyrus, as you can see here, frontal medial cortex, paracingulate gyrus, and inferior division of left lateral occipital cortex are known to be involved in drug cue reactivity. We also checked for differences in the whole brain negative neutral contrast between methamphetamine users and healthy controls. As expected, the parameter estimates were largely similar, except a few regions such as a right intracaudine cortex and right occipital fusiform gyrus. Before looking at brain cue reactivity, we need to look at results of behavioral tasks. As you can see, there are significant differences between commission and omission error rates, and also in reaction times between different blocks, especially between negative and drug cue blocks compared to neutral or blank cue blocks. So to look at differences in the brain responses involved in response inhibition between the 2 conditions of being faced with drug or neutral cues, we compared brain activations when individuals successfully inhibited their impulsive responses when viewing drug cues and their brain activations when they're viewing neutral cues. And so, as you can see on this slide, posterior cingulate cortex, left fusiform gyrus, inferior temporal gyrus, and superior parietal lobule had activation. And posterior cingulate cortex has also been strongly implicated as a key part of several inhibitory controls, like resisting the urge to take drug. So here you can see large clusters which show differential activation on posterior cingulate cortex and also the orbitofrontal cortex. And you can also see the results for the contrast between successful response inhibition when facing with negative cues and neutral cues. And there were some activations in inferior frontal gyrus and left medial frontal gyrus. And here you can see large differential activations across the occipital lobe, the orbitofrontal cortex, and the fusiform gyrus as well. As you can see here, the regions that showed differential activations during response inhibition in drug versus neutral and negative emotional versus neutral blocks are all involved in important cognitive domains that are relevant to both response inhibition and cue reactivity. So to conclude, our project adds to scan literature on brain activation differences during response inhibition when addicted individuals faced with different types of cues. There have been many attempts to derive clinically relevant biomarkers from response inhibition tasks and from the neuroimaging results of addicted individuals when they are performing response inhibition tasks. But relatively less attention has been paid to biomarkers that might be derived from analyzing the differences between response inhibition when addicted individuals are faced with drug cues compared to when they're performing response inhibition tasks under the condition of viewing neutral cues. And biomarkers derived from these differences might be more helpful in distinguishing different groups of addicted individuals from healthy individuals, and in predicting the treatment and maybe assessing response to treatment. So finally, I want to thank my co-authors of this study, Mr. Arshia Sangchuli, Mehran Zara Bidoki, Dr. Amir Hossein Batuli, Dr. Muhammad Ali Oghabian, and a special thanks to Dr. Hamid Ekhtiari for all his helps and supports. And thank you for your attention. I'll gladly answer any of your questions at the end. Thank you, Arshia.
Thank you, Amir Hossein. That was a fantastic talk. And after having had a look at Amir Hossein's presentation, we'll next move on to a project that's pretty different from Amir Hossein's in its scope and in its aims. Our next speaker is Sara Jaffakesh, PhD candidate in electrical engineering at Shiraz University of Technology. The project she'll be presenting is focused on an ongoing attempt at optimal region selection for biomarker development in the field of FDCR. Dear Sara, the floor is yours.
Yes. Thank you, Arshia. My name is Sara Jaffakesh. I'm a PhD candidate at Shiraz University of Technology. And I'm going to talk about feature selection based on fMRI drug reactivity test. As it mentioned in the last talks, we have some activations when the individuals face to drug use in the brain. And one of the most important challenges in the field is to select the regions that are reliable and have high absolute values. Traditionally, these selections was based on the absolute values, but the reliability is the most other important factor. And in this project, we set up the model that have both high level of activations and the reliability of the signal, and our subjects are 66 MET users and 22 opioid users. Both groups are male and are matched in age, education, and the other factors. And this is the block diagram of our model. First, we calculate the average signal of the whole brain, drug versus neutral contrast. And then the reliability of the signal was calculated. We select 14 brainstorm areas based on the literature and rank the absolute value of the signal and also rank the reliability of the signal. And with the combination of these two factors, we select our final regions as the winning regions. This is the activation map of whole brain drug versus neutral contrast. And if we parcel the brain in 246 regions based on Brainstorm Atlas and average the signal within each of these areas, and as you can see, we have some high activations in some brain regions, like ventromedial prefrontal cortex, inferior frontal gyrus. And also we have the activations in some regions like dorsolateral prefrontal cortex. These are all in the frontal cortex, which is very important in addiction. And also we have high activation in some other brain areas like temporal and amygdala. And as I said, the other important factor is reliability of the signal. And we calculate the reliability using intra-class correlation coefficients, ICC3K. And ICC3 is just a method to measure the reliability across multiple observations. And again, when we plot this in the Brainatome Atlas, we have this plot, and we can see many regions pass 0.5 threshold. And also, there are some regions that pass 0.7 regions in the frontal cortex and temporal cortex. And as Dr. Ikhtiyari mentioned, we have several networks in the brain that have an effective role in the addiction. And we choose 14 Brainatome areas based on these networks that some of them involved in executive network, some in salience network, and so on. And each of these areas composed of different brain regions. For example, DLPFC composed of I think 28 regions, and IFG 12 regions. And based on these areas, we search their reliability and absolute values within each 14 areas. First, we calculate the absolute values and rank the absolute values within each areas. We can see the above plot, the result of absolute values ranking. And the winning regions here we show with dark blue. We can see that some regions have a high level of absolute values in the frontal and temporal and amygdala. And the other factor was ICC, and we rank the areas with the ICC. And again, select the regions with the highest rank based on their ICC. Then we combine these two steps, and at the final step, we have the winning regions with a combination of absolute values and reliability. And as we see here, there are some regions that never have a high level of absolute value, nor very high reliability, but their combination has a very high rank, like DLPFC. And vice versa, there are some areas with high absolute values that not been in our final selection. And after selecting these 14 regions based on these method, we can use it for future analysis, like exploration as a biomarker. And the other works that we have done was to validate our method, for validate our feature selection with physiological and cognitive markers. We with physiological markers like heartbeat and breathing, because we know when addict people face to drug use, the rate of their heartbeat goes up, and their breaths deeper. And also we correlate them with cognitive features like self-reporting craving and the time that took for the people to rate their craving. And as we can see, our selected regions in most of the areas have a high correlation with these markers. And the other project that I'm working on is to predict the activations, drug activations, drug Q activations, using these regions with resting state connectivity matrix, as we talk about it now. Here is the flowchart of this method. After the selecting regions and selecting 14 winning regions, we calculate resting state connectivity matrix. And then we divide our subjects into training groups and testing groups. And using a regression model, we try to predict drug Q activations using this resting state connectivity matrix. And then we evaluate our prediction at the end using function connection relationship. This is the connectivity matrix of 66, the average of 66 met users. And the connectivity matrix is the connection between them in bold signal of these 14 winning regions. And this is the results of our function connection relationship. As we see here, our method has the ability to predict activations, drug activations in some regions like parahippocampus, hippocampus, and anterior cingulate cortex, and in some other regions, it has not very powerful prediction. And as a conclusion, we select 14 regions based on the reliability and the absolute value of the signals in MET and opioid users. And we validate our ROI selection by checking the correlation between selected ROIs with physiological and cognitive markers. And the chosen ROIs method can be used helpful in developing clinical biomarkers. And also, we use these winning regions to construct the predictive model to product activity based on resting state connectivity. And we observed that resting state functional connectivity has better ability to predict drug Q functions in some key areas related to addiction. And many thanks to my professors that helps me a lot. Dr. Kazemi, my supervisor, he really helps me and supports me in every situation, and he trusts me to be in this field. And many thanks to Dr. Ikhtiyari. I learn him every minute, and he really helps me. And also many thanks to Dr. Arabi and Dr. Erfroush. I'm so happy that I have a chance to work with this group. And our efforts lead to win the ISAM prize for 2 continuous years. And at the end, I wish our communication was not visual, and I could invite you in my very beautiful and historical city, Shiraz. And now I just hope that you all be healthy, and we can be your host in Shiraz very soon. Thank you for your attention.
Thank you very much, dear Sarah, for your fantastic and quite challenging talk, technically. Before I forget, I'd also like to note that both Sarah and Amir Hussain, our second speaker, have been awarded the ISAM Award for their presentations in this Congress. Now it's my pleasure to introduce our next speaker, Mehran Zarabi-Doki. Mehran is a medical student at the Shahid Sadeghi University in Yazd and is also a researcher with the Iranian National Center for Addiction Studies. His research focuses mainly on the potentials of FTCR in biomarker development, and he's already co-authored several papers and a book chapter in the field. He's also the Enigma secretary and has been involved in the projects you will hear about in the next 3 talks. Besides all that, he's been a very dear friend and colleague of mine for the past several years. His talk will provide an overview of the entire FTCR research space, discuss the many heterogeneities in the field and the potentials that the field has. Dear Mehran, we can have the presentation now.
Thank you so much, Arshia, for your kind words. As Arshia said, and I'm Mehran Zareh, and medical student and early career researcher, and I'm really honored to be in this panel. In this presentation, the results of an ongoing systematic review on the studies utilizing fMRI reactivity among different types of addiction are going to be discussed, along with some potentials for fMRI reactivity to be proposed as a potential biomarker. As you've seen in previous meetings, in previous presentations, there has been a huge progress during the last couple of decades in the understanding of the underlying mechanism of addiction, and many models were developed to explain them. One of the most reliable and validated ways that contributed to understanding what is happening inside the brain of a person with addiction is addiction-related cue reactivity, or drug cue reactivity, as most often said. This activates many regions and connection in the brain taking part in addiction, like attention, craving, self-control, and so on. So it was widely used to understand the dysfunctions in the brain in people with addiction. So we conducted a systematic review with this aim to gain a broad overview of the main information of the fMRI studies utilizing cue reactivity inside the scanner, or so-called FTCR studies. And for the second step, we were interested in more detailed methodological information about each study. A property that our work has is that it is pre-registered and readily available on the Open Science Framework, or OSF. There you can see the full explanation of the process of our systematic review, how the studies were included or excluded, the explanation of each item, and how the information of that is extracted. Here you can see the key terms, and here you can see the Prisma flowchart of our study. These are all the items that some were extracted and some are being extracted. As you can see, there are around 70 items, and in this presentation, the main extracted items are going to be presented. There were 331 articles in total. Studies were conducted in 18 countries, as expected, with the United States as the leading country, followed by Germany and China. You can see the number of studies done for each substance or behavior at the right. The studies done in each country are not completely homogeneous, and as you can see, the United States were more focused on nicotine, Germany on alcohol, and China and Iran on opioids. The first fMRI reactivity study among people with addiction was conducted in 1998, and as you can see, there is an overall increase in the number of studies each year after that. Among 331 articles, 225, meaning around 70% of the studies, were conducted in the last 8 years, and it shows the highly increasing interest of researchers to conduct their studies in this field. The most commonly investigated substance or behavior was nicotine, followed by alcohol, cocaine, opioids, cannabis, video games, methamphetamine, and gambling. Stimuli utilizing the fMRI scanner to induce reactivity among subjects were dominated by pictorial stimuli, around 69%, then videos, words, taste, auditory scripts, and others that you can see in the figure. You can also see that some types of stimuli were used specifically for one substance or behavior. For instance, a taste was only used for the alcohol. Another thing that we were interested in was to look at the studies which assessed the effects of an intervention with fMRI cue reactivity. There were 105 studies which did so, with most of them have a pharmacologic or psychotherapeutic intervention. As you can see, nicotine and alcohol are well addressed compared to the others, but use disorder in many substances like methamphetamine and cannabis have a wider space to be investigated by the fMRI cue reactivity. About the design of the studies, there was a relatively dominance of block designs compared to event, with 62% and 36% respectively. Also, only 14% of the studies used FDCR as a means to predict an outcome in the future. This outcome, for instance, can be relapse rate or addiction severity indices. Also, only 22% of the studies used more than one time points of fMRI sessions, and the interval between them was around 15 days on average. The presented results showed an overview of the fMRI cue reactivity studies among people with addiction, the general information and the gaps that should be addressed, and the foundation of the next two presentations on the enigmatic checklist was the things that we found heterogeneous in the field with this systematic review. Now, we're going to briefly discuss how fMRI cue reactivity can play a role as a biomarker. You've already seen some applications of the FDCR studies from two previous presentations. It definitely has a relatively long way to be validated as a biomarker, but there are many instances that FDCR could play a role as a biomarker, and I will quickly go over them to see different types of FDCR applications. So first, let's see what biomarker is. Based on FDA definition, biomarker is a defined characteristic that is measured as an indicator of normal biological process, pathogenic process, or biological response to an exposure or intervention. There are 7 types of biomarker that FDA has proposed: susceptibility or risk, diagnostic, monitoring, prognostic, predictive, pharmacodynamic or response, safety biomarker, and the last one is safety biomarker. And we are going to bring examples of FDCR studies among our database trying to play a role as a potential biomarker. First, let's start with diagnostic biomarker. A diagnostic biomarker is a biomarker which is used to detect or confirm presence of a disease or condition of interest, or to identify individuals with a subtype of disease. It is the first FDCR study ever. I brought this specific one to show that the first step in proposing FDCR as a useful tool was to show that it can suggest difference between two groups. In a study comparing 6 people with cocaine use disorder and 6 controls, this study showed that there are different activations between some regions in the brain when subjects are exposed to the cues inside the scanner compared to healthy controls. Another thing that this study did was of importance, that within those who had cocaine use disorder, they saw that there is a correlation between self-reported levels of craving and activations. Therefore, for these results for the first time, and that this could act as a potential diagnosis tool, though this study had only 6 subjects for each group, its novelty and proposing a new way of measuring dysfunctions in the brain of people with addiction led to 531 citations. Susceptibility biomarker is a biomarker that indicates the potential for developing a disease or medical condition in an individual who does not currently have clinically apparent disease or the medical condition. In this study, 43 college students underwent an FDCR task with alcohol-related cues, and then they were followed up for 1 year about use behaviors, and then they were categorized to 3 groups: those who remained moderate drinkers, those who transitioned to heavy drinkers, and those who remained heavy drinkers. Those participants who were susceptible of becoming heavy drinkers, but were not at that moment, had increased activation in reactivity to drug cues compared to neutral in the bilateral caudate, orbitofrontal cortex, medial frontal cortex, and inferior cingulate and the left insula in comparison with other groups. And so these results can probably work as a work to inform their prevention policies and to choose the ones who are more susceptible. The next type of biomarker is a prognostic biomarker and is used to identify likelihood of a clinical event, disease recurrence, or progression in patients who have the disease or medical condition of interest. In this study, researchers asked people with smoking who were willing to quit smoking to abstain for 24 hours and then undergo an FDCR scan. Then they were followed up for 7 days and then biochemically checked for the signs of relapse, and then they grouped them into relapse group and quit group. They found that people who relapsed in this period had higher activation in anterior cingulate cortex at baseline compared to quit group. Therefore, the prognosis of people with higher anterior cingulate cortex activation was worse. Prediction biomarker is a biomarker used to identify individuals who are more likely than similar individuals without the biomarker to experience a favorable or unfavorable effect from exposure to a medical product or an environmental agent. In this study, the researchers recruited people with alcohol use disorder. The subjects underwent an FDCR scan and then received a period of medication. Then they were followed up for drinking behaviors. And the results showed that those who had higher ventral striatal activation at the baseline had better response to naltrexone treatment, so it can be predicted prior to treatment. Because treatment is both money and time consumer, and these information can be used to choose best subject to be treated with a specific type of intervention, that this intervention is effective and is already predicted by the fMRI results. The other type of biomarker is pharmacodynamic or response, and is a biomarker used to show that a biological response has occurred in an individual who has been exposed to a medical product or an environmental agent. In this study, people with cocaine use disorder were grouped to either receive baclofen or placebo. They were then compared after the period of treatment, and it was shown that who received baclofen had less activation in mesolimbic network during drug cue reactivity compared to placebo group, and it shows how the brain response of the treatment is. And the monitoring biomarker is a biomarker measured serially for assessing the status of a disease or medical condition, or for evidence of exposure to a medical product or an environment. For instance, let's say that we know that there should not be a hypoactivation in the executive control network regions in people with addiction. And if there is normal activation in these regions, at least the cognitive control function of that participant is controlled. So when we're following up a person as a result of treatment, we should see that these activations are getting back to normal, or if they are going to be hypoactivated again, we should change our intervention as they are the signs of worsening the state of the disease or that our intervention is not working. I hope this presentation could give an overview of the heterogeneity in the field and also different ways of applying FDCR as a biomarker. My colleagues in the next talk are going to talk about how to conduct the validated biomarker in different aspects or not. At the end, I should especially thank Dr. Ekhtiari for his invaluable mentorship, my dear colleague Arshia Sanctuary for the collaboration, and also Hosna Tawakkali, Parnian Rafi, and Mohsen Ibrahimi for the great help. And thank you so much.
Thank you very much, dear Mehran, for your very substantive talk. I think it really captures the heart of what this panel is trying to be about. So as you can see, the FTCR field has a great number of potentials and is also highly heterogeneous across a number of methodological axes. The next two talks by Arash Khojasteh and myself will be trying to tackle will be about an attempt at tackling this issue in the field. The first talk is going to be by Arash Khojasteh, a medical student at the Mashhad University of Medical Sciences. He is one of the newest additions to our team and has already been involved in great work on a global consensus building effort in the field of FTCR. Dear Arash, the floor is yours.
Thanks, dear Ashia, for your great introduction and your kind words. Hello, everyone. This is Arash Khojasteh. Thanks to our great team, I am so honored to be here today presenting the methodological aspects of the checklist for fMRI drug Q reactivity studies, which was conducted in response to the challenges we are actually facing in the field. So in the following of what dear Mehran mentioned regarding one of the main current challenges in the field of FTCR studies, which happens to be methodological heterogeneities, we can see that this problem stems from different sources, such as the participants' characteristics, different types of cues used in the studies, also the duration of exposure to those cues, and the analytical methods as well. So, all in all, these items will lead to a state in which we will not be able to somehow conduct optimal comparative reports and meta-analysis regarding FTCR studies. But the question is, what are the possible solutions and options for facing these challenges appropriately? As you can see in the slide, some of the proposed solutions can be having pre-registered replicable protocols along published drug queue databases, also using data analysis guides and pre-registered and standardized analysis pipelines. But the one solution that we are going to discuss in the next couple of minutes is the extent checklist, which aims to act as a standalone tool for describing methodological details considered to influence results of FTCR studies. The checklist has been developed after a global expert consensus making processes, which has recently gone available on a medRxiv, and the link will be provided for you in the chat space in case if you're eager to take a closer look. As you can also see here, around a number of 60 scientists and colleagues from all around the world have collaborated for making this happen. And it was such an honor for me as a student to play even a small role in conducting this consensus. So in the next slide, we're seeing that let's take a few steps backwards and see how this concern was made at the first place and eventually got answered through its path. So the Enhanced Neuroimaging Genetics through Meta-Analysis Consortium, or AKA ENIGMA, is a network which tries to bring together researchers in imaging genomics to understand brain structure, function, and disease based on brain imaging and genetic data. Through this consortium, a group of scientists and researchers got concerned about the issues which was discussed before, the methodological heterogeneities in the field of FTCR studies, and Mehran briefly mentioned about that as well. So those people formed a subgroup within the ENIGMA, which was later known as the Addiction Q Reactivity Initiative, or ACRI, together as the ENIGMA ACRI Consortium, which is still open to new members who are interested in the field and is actually taking its future steps, which my colleague Ashia will discuss about it a little bit more later. So as you can see here, these professionals made up the core team in the consortium and led the whole Delphi study process regarding making the consensus with the help of others, which I'm going to discuss about the methodological aspects of the Delphi study in my next few slides. But what has actually happened through this consensus-making process was built on a structure of a Delphi methodology, a type of study which is usually consists of different rounds for making the consensus. That in this case, we had 3 phases of checklist development, revision, and rating. So in the checklist development phase, the steering committee, which I introduced you in the previous slide, they got together and made the initial checklist. And after the first round, they developed a basic set of categories, items, and their associated recommendations to report in actually FDCR studies. But before going through the checklist revision phase, the expert panel team was recruited based on the authorships and engagements in the FDCR studies, which were available in the systematic review, which Mehran discussed about. So these experts in the field were contacted via email, and they were invited to participate as a member of expert panel for providing their comments and notes about the initial checklist, which I told you about in the previous slide. They were also asked to suggest new items that they felt that it was overlooked. And also 10 of the steering committee members added their additional comments in this round. The steering committee members also processed the comments by removing repetitive, unclear comments along those outside the scope of the study. And they finally decided to apply or reject each note. The experts were lastly asked for their comments on the modified version before moving to the actually rating phase. So the rating phase, as the final phase, which was the final phase, participants from the steering committee and expert panel were asked to rate each item in terms of importance in the methodology of FDCR studies from a scale of 1 to 5. They also rated a yes or no answer for considering each of the proposed recommendations. So here you can see the general characteristics of the participants, which made up a good diversity in the field regarding different aspects such as countries, research fields, time spent in addiction research, and FDCR studies. In the current slide, you can see a part of the initial checklist, which was used in the development phase by the steering committee. This checklist was initially consisted of 5 main categories with 42 items, which was altered after the other rounds. And here is actually a snapshot of comments in the revision phase for a part which you may see 3 main categories and their associated items in the current slide. This might be a little bit unclear. In the next slide, yes, you can take a closer look about the commenting phase. And in the next slide here, actually here comes the rating phase of categories and main items to report, which was in a scale of 1 to 5, as I mentioned before, from the scale of not important to extremely important. It is a snapshot from the participants' characteristics category in this slide, actually. So, and for the recommendations, as I mentioned, it was a separate part. You can see that the rating phase was done by answering a yes or no question of whether having that specific recommendation along the proposed item or not. And this figure, this is actually the final outcome of the ratings for the main 38 items and 7 categories. You can see that the figure has depicted 2 thresholds of 70% and 80% for considering the item or not. You actually will discuss about the results in his talk in detail. And last but not least, this is the final checklist came out of this process of Delphi study design, which has the goal of acting, as I said, as a standalone tool for describing methodological details considered to influence results of FDCR studies. And in the next slide, you can also see another version of the checklist, providing the specific recommendations as well. Here for one of the categories, which is the participants' characteristics. Each category has had its own table for recommendations. And you can find the relevant instructions and the final checklist versions here in the OSF webpage. And actually, the manuscript also was collaboratively written by the steering committee and all the Enigma Acre Consortium. They also added their comments and revisions on the prepared document. And as my final words, let me remind you again that the Enigma Acre Consortium is looking forward to having new members as well for its future directions. I just here want to thank my dear colleagues, dear Dr. Ekhtiari for his mentorship, dear Dr. Mohaddes, dear Ashia Mehran for their great supports. I'm really honored to be in this team. I hope it finds you well. Thanks for your attention.
Thank you, dear Arash. So my own talk will expand on basically the conclusion and results of this consensus and checklist development study. I will make my presentation very brief because we don't have much time, and I do want to have a few minutes for the Q&A. So as you can see, the final checklist has 7 categories, 38 items, and 75 recommendations. In the paper, and I hope someone will provide a link in the chat, one of my colleagues, to the preprint on medRxiv, we extensively discuss the relevance of each category to the design of an FDCR study and how each category can influence and shape the methodological design and the results of the study. The first category was participant characteristics. These include items such as inclusion and exclusion criteria, basic demographic the information, and more advanced demographic information, psychiatric profiles, the handedness of participants, substance use profiles, and with more details such as route of administration for drugs which can be administered in several ways, and the pattern and severity of substance use, the usage of other drugs than the main substance investigated in a study, and abstinence status and addiction treatment status. As you can see, most of the items in this category pass the defined threshold for for very strict consensus, except for the ones that are marked with a dagger. The importance of most of these factors is very well investigated in the field. The importance of sex differences and the participant features, even things such as the menstrual cycle in female participants, is very well known. And the importance of treatment status and even motivation of participants and the phase of their you could say, addiction journey also is very relevant to brain activation patterns observed in FDCR. The second category is the general fMRI information. It includes items such as fMRI pulse sequence and other acquisition details, fMRI pre-processing pipelines and other details, fMRI data processing and fMRI data reporting. As you can see, all the items in this category passed the strict consensus threshold. As you can see, it's the one on the right. The less stringent consensus threshold is. This category also is quite important. We know that each of the items in the category can directly influence the obtained fMRI signal. For example, even head coils and the number of channels can have an impact. For example, pre-processing steps such as facial smoothing also can alter the results. The third category is the general task information, which consists of the general task design, number of task components, the requested engagement, which consists of the initial instructions given to the addicted individuals on how they should in fact engage with the task. Should they be passive viewers? Should they, for example, focus on how they feel about the images and so on? The temporal information of the task and its general temporal architecture and structure. As you remember, Amir Hossein presented a schematic representation of the temporal structure his task used. The order of blocks and events is also very important, and data and resource sharing in case the authors have shared their data is also important, and they're encouraged to in fact do so. And as you can see, all the items in this category, except for the last item, met the strict consensus threshold. And for example, one relatively recent study has shown that even the length for which cues are presented can have an impact on queue reactivity. The next category contains queue information, which consists of the sensor modality of queues. As you saw, queues in FTCR can be from a number of different modalities, and they can include taste cues, olfactory cues, pictures, audio cues, imaginary cues, textual cues, and so on. And it's very important to report what the exact sensor modality of the cues has been and provide justification for it if available. The source of the cues is also important. It should matter. It does matter whether the cues have been validated outside the context of the FTCR study in terms of their ability to, in fact, induce craving, for example, in the study participants. And also the content of the cues should be discussed. For example, for pictorial cues, it's important whether the content of the cues included drug use paraphernalia or was of drug using scenes, and so on. And it also should be mentioned how and whether, in fact, the drug cues and other cues, for example, neutral stimuli that have been used in this study have been matched for any factors, for example, their lighting, their visual content, and so on. The next category is craving assessment. As we know, one of the most important impacts of queue reactivity is that the task can induce craving in study participants. So it's very important to assess whether that has in fact happened. And the assessed craving can be used both to validate the task, and it can be included as a parameter in whatever model we build to analyze the data. And it should be mentioned, there's relatively little data on this, but it should be mentioned what the exact instrument was that was used for collecting craving data, and what the technology was. Was it a response box or a joystick or something else? Then craving assessment outside the scanner also is quite important, both before and after the task is conducted. And as you can see, there's been a recent review that has demonstrated that the instruments that are used in the this wasn't about specifically fMRI studies, but it does show that there are a great variety of instruments that can be used for craving assessment, and it's very important to discuss in detail what the tools used in the FTCR study have been. And the last category includes pre and post scanning considerations, pre-scanning training and familiarization with the scanner and with the scanning environment, pre-scanning drug and smoking consumption, whether it was allowed. It's important when the last time the participants were allowed to consume, whether their drug of choice or alcohol or cigarettes has been. And it's also important to note the other tasks and procedures in the imaging session and the order in which they were performed. And lastly, it's important for, at the very least, ethical considerations, whether there were provisions for participants to have a safe environment to reduce their craving after being exposed to the drug cues. And as you can see, abstinence and recent drug use can also greatly influence, as I said, FTCR, the FTCR signal. So the checklist has been developed after a global expert consensus making process, and it's available. I encourage you all to read it, whether you are planning to conduct an FTCR study, or if you just want to be able to read it, an FTCR study better, and to have a sort of a relatively objective tool to assess its reporting quality. And my talk was relatively brief, but this checklist was in fact presented in much more detail and in a talk involving many of the experts who were involved in developing it and in writing the manuscript. You can view the talk that in which this checklist was discussed on a YouTube channel, and I think my colleague will share the link to the video. Thank you very much. Now I'll hand the mic to Dr. Ikhtiyari for a few minutes, so you can have a conclusion, and then we'll move on to the Q&A.
Yeah, I'm really happy that we had such a kind of series of nice presentations today. I'm really happy that these young, brilliant minds are going to lead this field. They are already doing a lot of really good things. I'm sure that they would be leaders in this field in the next couple of years. Let me just give you an overview about what we have discussed so far and potentials, how we might be able to do collaborations. What are the areas that we need others who are interested to collaborate and join us in different projects that we are thinking about. Meanwhile, so what we can do while I'm presenting the ideas for future collaboration, how others might be able to join us in this collaborative initiative, would be having questions and taking questions in the chat space, so we can also have an idea about the number of questions that we have from audience. Then we can have a balance between the discussion and concluding remarks and also the questions. So for the areas for future collaboration, first of all, as we have discussed today, we do fMRI drug cue reactivity. We have neutral cues and drug-related cues, and we show these cues to the people inside the scanner. So definitely we need to have pictorial databases. So we need to have pictorial databases that are strong enough to elicit these activations inside brain. This is what we have started to do from 2003. These figures are from Iranian National Center for Addiction Studies, where we have been developing these figures. I clearly remember the challenges that we have to develop these figures and photos and putting them on printed sheets and put them mounting them over the wall and asking subjects to rate them. So developing new cue databases is definitely one of the really important areas in this field. There are already some new published databases that people have developed, but there are still significant needs for these databases. We have published a database with 120 methamphetamine and 120 opioid cues, and also neutral cues. And this is one of the largest databases in the field, but there are still needs for other areas. And those who are interested to collaborate and join us to develop new databases and also test the database that we have developed in, for example, Iran. For example, we have a group of colleagues in China who are replicating what we have collected in US, and there are a group of people who are doing the same in Iran. So thinking how we can have these databases validated in different contexts, in different countries, also having new databases. So that is one area for collaboration. The other area that, as you have seen in talks by different presenters today, when we have fMRI drug cue reactivity, we have activations in different areas in the brain. And the question is, okay, how we can separate these areas in terms of different functions that they are corresponding. And as Sarah beautifully showed us today, we can put these activations in different, let's say, brainotome ROIs or regions of interest. We have 246. We have an atlas that we call Brainotome Atlas that has 246 parcels within the atlas. And if we just put the activations in these 246 areas, you can see this nice figure of having different activations in different parts of the brain. And these are the abbreviation of those brain areas like superior frontal gyrus or middle frontal gyrus. But beyond that, how we can have other derived measures, how we can measure, how we can clusterize these activities to several clusters, that each cluster would correspond to something like memory, executive function, self-referential processing. Or beyond the activations, how can we have different networks, how these areas are interacting together while we are exposing people to drug-related cues. So we need to have more derived measures from these, let's say, raw brain activation. So that is another area for future collaboration. The other area for future collaboration is about time, because we expose people to drug cues several times inside the scanner. So we do repeated cue exposure. And one of the questions is, okay, if we expose people to drug-related cues several times, how dynamically their brain would respond to these temporally presented drug cues. And we have done different studies in terms of, for example, if we expose people to drug cues between block 1, 2, 3, 4, there are areas that they do not have significant changes over the time. So for example, areas like inferior frontal gyrus, whether it's left or right, they still have significant higher activation in drug cues compared to neutral cues, as you see in this figure. So they still keep their activation, and there is no temporal dynamic, sort of temporal dynamic response, whether it's habituation or sensitization in the activation. But there are certain areas that we have, as you can see here, a level of habituation. So when we expose people to drug cues, the level of activation would just go down in repeated exposure. And the question is why, and what are the clinical significance of these different temporal dynamics in different brain areas, how we can explore more in terms of the temporal dynamics of brain activation. So that is another really important area for future studies and future collaboration. But definitely we are interested to have students from mainly computational science to explore these temporal dynamics. The other area that is critically important for us in future would be how we can replicate the results that we have in different countries around the world. So when we have a discovery sample of, let's say, having this specific pattern of activation within ventromedial prefrontal cortex, if we have another replication sample, could we have the same pattern of activation? And if we go for the second replication sample, and even the question would be, if we do the same exposure within the same database, within the same group of participants, and having replication being retested, and do we have the same activation? As you can see here, for example, we had the same activations within the sample 2, but when we exposed them again to the drug cues in retest, we didn't have that pattern of activation. So that is quite important in terms of exploring these replications. And we have done that recently, and there are publications according to that replication, but there are still many areas for future collaboration on replicating what we have developed. The last area for future collaborations would be thinking about how we can develop interventions to modulate this drug cue reactivity. Trying to think about, okay, if attention is involved, if memory is involved, if interoception is involved, how we can develop specific cognitive interventions to modulate these certain cognitive functions, and then do imaging before and after these interventions. If we give people information about how your brain is processing drug-related cues, can we pick up the changes in brain activations by these even short interventions? Can we have different patterns of activations with having these interventions? And as I have mentioned, that these interventions could even extend to areas like food cue activity, and then think about how we can have these interventions within what we call basically drug cue reactivity. So that is another area for collaboration. Right now, we are developing new interventions in combining cue exposure with what we call episodic future thinking and memory reconsolidation, developing new interventions. I'm working with Parnion right now on that idea. And those who are interested to join us for those projects, we definitely would be happy to have you on board. Okay, so just giving you a wrap up, we have discussed that there are potentials for developing tasks, thinking about how we can develop new measures, network level measures, different levels of measures, and having pre-registered analysis pathway, as Sarah mentioned, in terms of making sure that we are looking into areas that are gonna be more reliable and have significant activations, seeing how we can find reliable and replicable results. And as Mehran has mentioned, thinking about validity, whether it's diagnostic, predictive, or treatment monitoring validity, there's another really important area in this field, and then developing new interventions. I just was interested to share one of the photos that we had from the group of colleagues that we have been working together something like 15 years ago in the Iran National Center for Addiction Studies. And yeah, I realized that without having collaborative partnership and doing teamwork, it is really complex to push forward and move for for future studies. And this is my last slide. This is one of the posters I found recently. It goes back to 2007, exactly 13 years ago. And this workshop was about how we can do neuroimaging for the first steps in Iran. And those who are presenters here, they are from wide range of different disciplines. We have people from psychiatry, physics, biophysics, computational science, computer science, electrical engineering, and all these people are collaborating together. That is the beauty of neuroimaging and functional neuroimaging. And we definitely encourage those who are from different disciplines to join us. And there is always a specific space for those who are interested to science to work with us in the field of fMRI drug reactivity.
First, let me thank you for your presentation, and I think you made a great wrap up out of what is discussed in this 1.5 hour. Actually, we don't have a specific question in the chat space here, but I'm not sure if we have much more time to ask the audiences to ask the questions lively with their videos or voices. I think we have received just actually a question right now. As you see, Zahra Abbasi has asked the question, can we work on prevention of addiction with neuroscience? That's a general question, I think. Thank you for your question. Would you like to answer this question, Dr. Ikhtiyari?
We had the panel on neuroscience for addiction prevention on Friday, and the panel is being recorded. So hopefully we will circulate the video of that panel. So we discuss in details about how neuroscience could inform addiction prevention. And there are definitely lots of great potentials for that. So I highly recommend you to see that video. That would definitely be helpful.
Thank you very much for this. Actually, general question, I think a comprehensive answer would be needed, and that would be the best answer to take a look at that presentation. Okay.
Thank you, everyone, for attending this talk, and I'd like to thank the other speakers on this panel for their fantastic talks.
Actually, I just want to encourage everyone to start this collaboration, this emails, texts, or something like this. I myself started just like with sending an email, something like 3 years ago to Dr. Ikhtiyari, and it started very nice, with very nice answer. And at that time, I started my collaboration with this team. I actually encourage you, don't hesitate, and start collaboration, start connection with this team. It would really worth doing. Thank you very much. I think we can say goodbye to everyone. Thank you for joining, and thanks for all the presenters.